Starting with the first half of the topic, endoplasmic reticulum. Endoplasmic reticulum is abbreviated as ER. ER is a double membrane system found throughout the eukaryotic cell. The membrane is continuous with the nuclear membrane and the plasma membrane. ER is absent in prokaryotic cell. George Pallet in 1974 made electron microscope studies of the subcellular fractions in demonstrating endoplasmic reticulum and Golgi complex. In course of staining the cell with basic dyes, it has been observed that certain regions of the cytoplasm show intense staining. These regions were termed ergastoplasm in the late 19th century. Later, through fine sectioning, it has been established that the largest membranous network in eukaryotic cell is the endoplasmic reticulum, consisting of tubules, vesicles and sacs. Endoplasmic reticulum, that is ER, is found in two forms such as granular or rough endoplasmic reticulum, abbreviated as RER, and an agranular or smooth endoplasmic reticulum, abbreviated as SER. Ribosomes are attached with the RER, which cause certain regions of the cytoplasm to stain with basic dyes. SER lacks ribosomes. The smooth endoplasmic reticulum, that is SER, is the region where the synthesis and metabolism of fatty acids and phospholipids occur. Different cells may contain different amount of SER depending on their nature and function. Enzymes that modify toxic chemicals or carcinogens to reduce its toxicity are present in SER. These toxic chemicals are secreted out of the body through the formation of vesicles by SER. Under the electron microscope, the SER consists of an interconnected series of convoluted tubules. In addition to the presence of ribosomes in the rough endoplasmic reticulum, that is RER, it appears as large flattened sheets of membrane showing lumen or central cavity under the electron microscope after thin sectioning of the cell. RER is present in cells in high frequency which participate in the synthesis of protein for export. The ER is composed of following three kinds of structures, that are cisternae, tubules and vesicles. Cisternae. The cisternae are broad, long, flattened, sac-like unbranched tubules having the diameter of 4 to 5 nm. They are arranged parallel in bundles of stacks to form lamella. They are interconnected with each other. The cisternae usually occur in those cells which have synthetic activities as the cells of pancreas, liver, plasma and goblet. Tubules. The tubules are branched structure forming the reticular system along with the cisternae and vesicles. They appear as circles in electron microscope, that is EM sections. The tubules are more diverse in shape and having diameter of 5 to 10 nm. They occur almost in all cells. They are most common in the synthesis of steroids and in the pigmented epithelial cells of retina. Vesicles. The vesicles are oval, membrane-bound vacuolar structures having the diameter of 2.5 to 50 nm. They often remain isolated in the cytoplasm. They occur in most cells. Now, we will learn the functions of endoplasmic reticulum. The polymorphism of the ER in a variety of cells and its changes with differentiation and cellular activity have made very difficult an analysis of its multiple functions. Some of the generalized functions are as follows. Mechanical support. The ER along with the cytoplasmic matrix participate in the mechanical functions of the cell. By dividing the fluid content of the cell into compartments, The ER provides mechanical support for the colloidal structure of cytoplasm. Membrane fluidity, flow and circulation. The ER may act as a kind of circulatory system for intracellular circulation of various substances. Membrane flow may also be an important mechanism for carrying particles, molecules and ions into and out of the cells. Membrane flow is active between ER and nuclear membrane. This flow provides explanation for export of RNA and nucleoproteins from the nucleus to the cytoplasm. The membranes of ER at body temperature is highly dynamic and also fluid. The bound ribosomes are mobile on the membrane and their mobility is controlled by its fluidity. The concept of flow refers not only to the content of the ER but to the membrane proper. Transfer of secretory proteins is accompanied by a flow of newly synthesized membrane proteins that are incorporated into the rough ER, that is RER. Transport across ER membrane. ER has enormous internal surface, about 11 meters square per ml in liver cells, which is important for exchanges taking place between the matrix and the inner compartment. The vacuolar system has osmotic properties. Diffusion and active transport may take place across the membranes of ER. The presence of carriers and permeases has been postulated. The vacuolar system separates the cytoplasm into two compartments. This makes possible the existence of ionic gradients and electric potentials across these intracellular membranes. This has been applied to the sarcoplasmic reticulum, a specialized form of ER found in striated muscle fibers, which is considered as an intracellular conducting system. The sarcoplasmic reticulum is involved in the concentration and storing of calcium ions. When the muscle is stimulated, the calcium ions are released, resulting in muscular contractions. Membrane biogenesis, assembly and recycling. ER membranes may develop by evagination from the nuclear envelope. At telophase, the nuclear envelope is reformed by vesicles of the ER. There is no de novo synthesis of membranes. They grow by expansion of pre-existing membranes. The cell receives a full set of membranes from its ancestor cell. Membrane biogenesis assumes a multi-step mechanism involving the synthesis of a basic membrane of lipids, intrinsic proteins and addition of enzymes, specific sugars or lipids. Bulk of the intracellular membranes are synthesized in the RER and then transformed into the membranes of the smooth portion and the Golgi complex. The portions of membrane move in two general directions. 1. From the ER through the Golgi complex to the lysosomes or peroxisomes or plasma membrane, and number 2. From the plasma membrane to the lysosomes and Golgi complex. These movements represent a dynamic process. The membrane is recycled back and forth between the cell surface and the interior. Formation of microbodies. Microbodies are closely related with the ER and are in connection with the ER cisternae. The microbodies are formed as dilations of the ER. They are rich in the enzyme peroxidase in peroxisomes, catalase, and the amino acid oxidase. In plant cells, these are called glyoxysomes because they contain enzymes of the glyoxylate cycle. Moving ahead, we discuss the special functions of smooth endoplasmic reticulum, that is SER. Detoxification. An important function of SER is in the detoxification of many endogenous and exogenous compounds. The prolonged administration of certain drugs produces an increase, that is hypertrophy, in the SER and the induction of the specific enzymes. In the liver, the SER is specialized for the detoxification of a wide variety of organic molecules, including barbiturates. At least two complex series of enzymes that oxidize these substances are present in SER membranes. Lipid synthesis and storage. SER is involved in the breakdown of fatty acids and in the conversion of fatty acids to acyl coenzyme A. The SER has been associated with the storage of lipids. Synthesis of cholesterol and steroid hormones. SER is rich in cells engaged in lipid metabolism and in those producing steroid hormones. SER is the site of cholesterol synthesis and storage. In the testes, ovary and the adrenal cortex, the SER is involved in the synthesis of steroid hormones. Glycogenolysis. SER is related to glycogenolysis. SER is engaged in the secretion of glucose to the exterior and also involved in its protection from the action of glycolytic enzymes in the cytoplasmic matrix. Thus, it regulates the pathway of glucose metabolism in the cell. The withdrawal of glucose from glycogen deposits is mediated by the SER by way of glucose 6-phosphatase. This enzyme catalyzes the dephosphorylation of glucose 6-phosphate to form glucose. The function of the glucose 6-phosphatase in the SER membrane is to catalyze the removal of the phosphate group and transfer the glucose formed into the space within the SER. From there, the glucose enters into the bloodstream from where it can be transported to needy cells. Ionic gradients. In striated muscle cell, there is an extensive, highly specialized SER called the sarcoplasmic reticulum, which is involved in carrying the stimulus from the nerve cell into the cytoplasm of the muscle fiber. SER is a delicate plexus surrounding the myofibrils. The longitudinal sarcoplasmic tubules form terminal cisternae. Photoreception. The myeloid bodies in the pigment cells of the retina are modified SER. It is made up of a number of tubules packed over one another. Myeloid bodies are involved in photoreception. Furthermore, we detail the special functions of rough endoplasmic reticulum, that is RER. Function of signal sequences and protein processing in ER. The association of mRNA ribosome complexes to ER is done by the signal sequences located near the N-terminal end of the protein. It directs the protein synthesis. This is known as signal hypothesis. The actual mechanism of the signal sequences and protein processing in ER have been studied in case of seed storage proteins of maize and pea. Mechanism of translocation of peptides through the membranes of the ER. After synthesis of proteins in ER, there are several types of processes for transfer of proteins into and through membranes of ER, mitochondria, etc. Generally, peptides with clear and unclear signal sequence are transported into these membranes. The transport of protein takes place along with its translation, so it is called co-translation process. This transport of peptides takes place either after cleavage of terminal signal sequence or it may enter into two domains, that is, in the cytoplasm and in the lumen of ER with the cleavage of signal sequence. The signal sequence remains bound to the membrane. Transport of various protein molecules. RER, that is portion of ER bound with ribosomes, takes part in the synthesis of certain specific proteins. These proteins are glycoproteins, lysosome proteins, membrane proteins and some organelle proteins. These proteins synthesized on RER transport into the cisternae or space of the endoplasmic reticulum for exportation. The proteins synthesized on free ribosomes are utilized within the cell. For example, the protein ferritin synthesized on the free cytoplasm ribosomes of the liver cell is retained within the liver. The other proteins, albumin and gamma globulin, are synthesized on the membrane-bound ribosomes. These are then secreted into the blood.